Introduction.
Good morning, teacher.
Good morning, students. Today we learn plant growth and development. Who will tell me what is growth in plants?
Ma'am, increase in the size of the cell and elongation in them signifies the growth of plant.
Very good, Suresh.
Ma'am, can we measure the growth of plants?
Good question, Rahul. Yes, we can. The growth of a plant can be measured by considering various factors such as nutrients availability, water availability, oxygen, etc. One more thing I would like to tell you that there are 5 plant growth regulators. They are auxins, gibberellins, cytokinins, ethylene and abscisic acid. Okay, now it's time to learn about plant growth and development. Objectives. At the end of this lesson, you will be able to explain growth in plants, explain phases of growth, define growth rates, to know quantitative comparison of growth, explain conduction for growth, define differentiation, dedifferentiation, and redifferentiation. Define development, explain plant growth regulators, describe photoperiodism, describe vernalization. Growth. An irreversible increase in the size and number of the cells by division and enlargement is called growth. In plants, the growth generally is indeterminate. Plants have the capacity of unlimited growth because of the presence of meristems at certain locations on their body. The cells of meristems are capable of dividing by their own. When new cells are added in the body of the plant due to the activity of meristem, it is called open form of growth. The root apical meristem and shoot apical meristem are primarily responsible for the elongation of the plant along its axis. In dicot plants, the lateral meristems, vascular cambium, and core cambium are responsible for secondary growth of the plants. Growth is measurable. Growth of the plants can be measured by various factors: increase in cell numbers, fresh weight, dry weight, size of the cell, etc. For example, watermelon cells can increase in size up to 350,000 times. Increase in surface area shows the growth of leaves. Phases of growth. The period of growth is divided into 3 phases: meristematic, elongation, and maturation. Meristematic phase. This phase has continuously dividing cells and is restricted to the apical meristems, both at the root and shoot tips. In this region, the cells are rich in protoplasm. They have large nuclei. Phase of elongation. The aim of this phase is enlargement of cells. Phase of maturation. In this phase, cells undergo maturation to accomplish their final size, shape, and structure. Growth rates. The increased growth per unit time is termed as growth rate. Plants show 2 types of growth: arithmetic and geometric. Arithmetic growth. Only one daughter cell continues to divide while others become mature. For example, root elongating at a constant rate. Mathematically, LT is equal to L0 plus RT, where LT is equal to length at time t, L0 is equal to initial length, R is equal to growth rate. On plotting length against time, a linear curve is obtained. Geometric growth. Initial growth is slow, followed by a rapid increase in growth. followed by a phase where growth slows down. All cells, tissues, and organs typically show this type of growth. Mathematically, W1 is equal to W0 into exponent RT. W1 is equal to final size. W0 is equal to initial size. R is equal to growth rate. T is equal to time of growth. E is equal to base of natural algorithms. Quantitative comparison of growth. Two ways to compare growth quantitatively are absolute growth rate and relative growth rate. Measurement of total growth per unit time is called absolute growth rate. An absolute increase is shown in the areas of the leaves A and B to form leaves A1 and B1. Growth of given system per unit time is called relative growth rate. In the given figure, both leaves increase by 20 centimeter square, but a relatively greater growth has occurred in leaf A. Conditions for growth. The conditions necessary for growth are nutrients. Enough supply of nutrients is necessary for synthesis of protoplasm and act as a source of energy. Water. An adequate supply of water is essential for growth. It maintains cell turgidity, which enhances growth. It also provides medium for enzymatic reactions. Oxygen. It is vital for respiration and for release of energy. Temperature. The most desired temperature for growth is called the optimum temperature, and it ranges from 28 degrees to 30 degrees Celsius. Temperature above 45 degrees Celsius coagulates and damages the protoplasm and stops growth. Differentiation, dedifferentiation, and redifferentiation. Differentiation is the process by which meristematic cells undergo changes in the structure, shape, to perform the specific functions. For example, after the DNA synthesis of the protoderm cells, cell expansion takes place and it forms immature trichome. After that, nuclear migration takes place and then branching occurs and then it reaches to maturation stage. Dedifferentiation. Dedifferentiation is the process by which the already differentiated or mature cells get back the power of division and become meristematic. Redifferentiation. Redifferentiation is the process by which the meristematic cells formed by dedifferentiated cells undergo changes in the structure and shape to perform the specific function. Development. Development is the series of the changes in which organisms undergo in their way from the embryonic state to maturity, that is, from lower state to higher state of organization. Plants undergo different pathways in phases of life in order to form different kinds of structure. This is termed as plasticity. If different shaped leaves present on the same shoot or on different shoots of the same plant, it is called heterophyly. For example, water crowfoot, water startwort. Plant growth regulators. Characteristics. In all the plants, there occurs minute quantities of certain chemical substances that regulates growth and differentiation. These are called growth regulators. A growth regulator may be defined as an organic substance produced naturally in plants controlling growth and other functions at a site remote from its place of production in minute quantities. These are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. The discovery of plant growth regulators. In the last 60 years, a large number of growth regulators have been isolated from plants. The first indication of their existence was given by Darwin 1880